Dr. Fahida. Fahida, can we start?
Okay, ma'am.
Okay, please.
Good afternoon to all. Warm welcome to the 10th day of Golden Jubilee Lecture Series, hosted by the Department of Seed Science and Technology, College of Agriculture, Velanikere. First of all, I invite Srimati Fahida PA, Assistant Professor, Department of Seed Science and Technology, to deliver the welcome address.
Good afternoon, all. Under the Golden Jubilee celebration of Kerala University, today is the 10th lecture of the online lecture series, is Sustainability, Caring, and Serving Spirit for a Self-Secure Future, organized by the Department of Science and Technology, College of Agriculture. It's a great honor to welcome the honorable speaker of the day, Dr. Anjali Anand, Principal Scientist, Division of Plant Physiology, Indian Agricultural Research Institute, New Delhi. The topic of the lecture is boost stress tolerance to seeds through physical seed treatment. We are very blessed to have Dr. Anjali Anand to deal with this topic, who has vast knowledge and research experience and a well expert in the seed physiology, climate change, and agricultural physiology. On behalf of all the participants in the Department of Science and Technology, I extend a warm welcome to you, madam, for the 10th lecture of our lecture series. It gives me immense pleasure to welcome Dr. Rosemary Francis. She will be coming. Was actually organized and scheduled all this lecture with all experienced personalities in the field, and she is always with us for a successfully organized program. On behalf of all of us, we welcome you, madam, for this. Now we welcome Dr. Vijay Bastin, Professor and Head, Department of Science and Technology, who has taken all the efforts in organizing to this lecture. And I am so happy to welcome Dr. J. Krishna Kumar, Assistant Professor from Department of Plant Physiology, College of Agriculture, Velanikere. And I am so happy to inform that he is the student of our honorable speaker in IARI and who willingly accepted to offer the concluding remarks after this session. I also take this opportunity to welcome Ms. Ruth Olivia, who is introduced as the speaker of the day, and Mr. Vaishak, a MS student of our department, who is offering to give the vote of thanks after the session, and also all the other students and staff of the Department of Science and Technology who are sincerely supporting in all the activities in organizing this webinar. I also welcome all the participants, including teachers, scientists, and students from in and outside the university who is attending this webinar with utmost interest from beginning to the end of this lecture. And I'm sure that this lecture will be very useful and request to all the participants to use the time maximum and interact with our honorable guests. Thank you all.
Thank you, ma'am. Now I invite Ms. Ruth Olivia for introducing the speaker.
Thank you, Vaishak. A great honor to introduce our present speaker, Dr. Anjali Anand, who is born in the year 1968. She is specialized in climate change, seed physiology, and abiotic stress physiology, and working as a principal scientist. She has completed her graduation from the Delhi University and her master's and doctoral programs in the field of plant physiology from IARA New Delhi. She has many contributions in the advancement of science, and some of them are in the development of protocols for the static and pulsed magnetic field mediated dry seed priming for improving the seed vigor in various crops, and found that vigor improvement is mediated through ROS and RNA signaling, and also the role of ROS in magnetopriming mediated stress tolerance of various crops. Also, the findings like the role of amylases in nocturnal starch breakdown in rice under high night temperatures, and also the high night temperatures reduces the spike fertility, yield, and grain quality in rice has been contributed by the man. She is also a part of many research publications and the editor of some of the books in the field of plant physiology. She's also achieved many awards like Fellow of Indian Society for Plant Physiology, editor as an editor for Indian Journal of Plant Physiology, Best Paper Award by Society for Advancement of Human and Nature, and also Fertilizer Association of India. And she has an achievement of outstanding academic performance during her MSc and outstanding presentation of research contribution from IRA. She is also a part of many projects like Impact, Adaptation and Vulnerability of Indian Agriculture to Climate Change Information Needs and Researchable Issues from 2004 to 2013, and Electromagnetic Energies for Biostimulation and Post-Harvest Preservation of Seeds and Agri-Products from 2007 to 2011. Impact Assessment of Climate Change on Major Crops and Integrated Vulnerability Assessment of Agriculture in India from 2009 to 2011. Magnetopriming of high-value vegetable seeds for invigoration and productivity enhancement from 2015 to 2019. With this, I want to invite you, ma'am, to the present lecture.
Thank you, Ruth. Thank you so much. Very good afternoon to Ms. Fahida, staff, attendees, and dear students who are present for today's online lecture. And I'm happy to be a part of your Golden Jubilee celebrations, though it is online. And I'm also very happy that two of my very good students are here. Krishna and Sini also is here. I think she's posted in the regional station. But she was here. She was in fact a MSc student. And today I'll be presenting her work also. So I'm very happy to see her after so many years. And I know you would have celebrated this program in a grand way had the COVID not been there. And I would have in person visited you all, seen your laboratories, met you people, and looked for further collaborations with all of you. Kerala being a very beautiful place. It's also known as heaven. So as a tourist, I have often visited Kerala, but I would have really loved to visit the university also. So my today's topic is, can I share my slides? Just give me a minute. Are my slides visible now?
Oh, man.
Just a minute. We have most of our meetings on Zoom or MS Teams. So I'm not very well versed with Google Teams. So I just open my presentation and share the screen, that's it.
And there will be an icon where it shows present now. You have to click that and then you will have to present.
Okay.
At the top of the screen, you can see three dots. Left to that, there is an arrow mark type of icon.
Yeah, I have shared that, but it is showing me this share your entire screen.
Share entire screen. They have to click the share entire screen.
Okay, share.
Not share. Yeah.
Of treatments. Then I will take up the physical energies which are being used for seed priming treatments. And the third will be the effect of these energies at the plant level. I'll be showing you some field trials that we have conducted ourselves. And then I will move to the magnetopriming part where we specialize, our group specializes. and I'll be telling about the stress-related experiments that we have done with respect to magnetopriming. So I start with a picture of despair, but there's always hope after despair. And this despair has also been created by mankind. So it is mankind or we people who have to look for solutions to overcome the adversities of climate change. And climate change has become a reality. And being able to understand seed priming because it is a part of your course. Just to start with my talk, what is seed priming? It includes all the post-harvest treatments that you give to the seed that improves in germination, followed by seed growth. It gives seed to the seed, and it also helps in delivery of all the economic practices or packages that can be realized for getting a better yield from the same which has been treated. So the potential crop can be realized through in terms of seed. Then this is not waste free, but this is waste free. Then this is suitable for organic farming. It has been said that since you are not using any chemical treatment for seed priming or later on, so it can be implemented with other organic farming techniques, all the package of practices that you're using for organic farming. And of course, both the ROS is involved in the signaling process. So the hormones which are involved process, primarily GA and ABA, their levels are changed. So GA from literature and also from papers, most of the students must be aware of it. It is involved in the germination process. So it has been seen that there is synthesis of GA and there is a catabolism of ABA. So ABA, which was accumulated during the seed maturation process, its level goes down by the upregulation of the catabolic genes, and the ratio becomes in favor of GA to ABA. Then one study has also reported that there is biogenesis of mitochondria because seed germination requires a lot of energy so that by the mitochondrial process and respiration is increased. So for that, there is multiplication of the mitochondria within the cell when the seed is put for immunization. Besides that, there are epigenetic changes which have been reported. There is DNA repair mechanism which is occurring. The aquaporins are stimulated or induced because aquaporins are the channels through which the water is going to move in the seed. Then there's a down regulation of the late embryogenesis proteins. Then the alpha amylase protease, which are involved in the breakdown of starch and proteins, they are also upregulated. Their activity increases when the seed is germinating. Metallothioneins are another class of proteins which also act as antioxidants. So they are also very good scavengers of the ROS, and they help in maintaining the ROS levels within the oxidative window. Then HSPs, besides their role in the induction of the protein during the high temperature tolerance, they are also constitutively produced, and these are also related to many other processes. They occur in the signaling cascade, so their levels are also seen to be increased when the seeds are put for priming or later after priming when they are put for imbibition. So this is a figure which shows that the uniformity in germination is there. If you see the radicle, you'll see most of them are of the same size. So the seeds have been primed and they have reached to the same level. Now when they are put for the germination, you see similar response in most of the seeds. Whereas here there's a staggered kind of a response. Few are germinating from the same seed lot, and few are ready for the radicle to come out. So whatever story I have just now told you, it sums up to that priming will lead to the early vigor in the seed and a uniform population of the crop, which is the seed of which is being primed, and it thus improves the seed quality. Then it reduces the seeding rate because you are getting one plant from each seed that is sown. That is in the best scenario. Then, because the seed have a head start, they have started early when they have been put for germination in the soil, so there will be earliness to flowering, and it also increases the yield under stress. And here I talk about the soil related stresses, which I had shown in the second slide, that is drought, salinity, heavy metal, etc. So that kind of stresses are also taken care of when the seed is primed before being sown in the field. So this term early vigor you must have come across in your breeding classes also, genetics and breeding people. They are using it as a very important trait for selection of a genotype under stress conditions. So here also what we are doing by priming is we are introducing early vigor in the seed. So this is a field trial in which you can see that this was a low early vigor line. So early vigor was less in this. But in this line or this germplasm, early vigor was high. So when they have been put under water stress, so this had an ability to pick up or revive or reclaim compared to this one, which was a low early vigor line. So early vigor trait, which is being used by breeders also, it helps in faster leaf development, plant canopy development will be faster. Then it will reduce the soil water loss because once the canopy is established early in the field, it will not allow water to be evaporated, and it will also be a very good competitor against the weeds because it will pick up faster than the weeds. Then it will improve the nutrient uptake, and finally everything comes down to high biomass and grain yield because our main goal is to have high biomass and grain yield. This becomes a very good technology and is widely being used for vegetable seeds. More than field crops, it is being used for vegetable seeds, both in the field production and also in the greenhouse production. So at both the places, it has its different advantages. In field production, I have just enlisted the advantages that it was that it was reducing the seed rate, there was less requirement for thinning and tolerance to germination. And in vegetable crops, it's very important because your crop is uniform. All the plants have come to the same level. So most of the crops have manual picking. So a laborer or a farmer who goes into the field to pick up or harvest the crop, he can have maximum harvest on the day of the picking. So this is a very good advantage. This will happen in this case also, but I am putting it under field production. In case of greenhouse conditions, because you are taking a head start, as you see in a relay race, the person who takes the head start has more chances of winning the relay race compared to the other players who are a little behind. So our crop here has taken a head start. So in the same area, you can thus have, even if you save a week with one crop, you can have more crop in the same area throughout the year. It also reduces the heating cost because it reduces the crop duration, which happens because of the priming treatment. This I've just picked up from the internet where many companies abroad, they are selling their seed priming agents. Of course, they're not disclosing what the agent is, whether it is hormonal, chemical, but they are selling it for the production of vegetable crops. And here they have advertised it that it can um, give a very good yield under a treatment compared to the untreated crop. This is a field from Zimbabwe where maize crops. On the left side, if you see, the crop is very tall. On the right hand side, it is a bit shorter. So in this case, the seeds were primed prior to sowing. So the difference can be seen in the growth of the crop. Here also at the germination, if you see, here the treated seeds or the primed seeds has emerged faster than the unprimed seed. So this was about the two different kind of priming treatments: invasive, non-invasive, or you may call them conventional, non-conventional, or wet priming, dry priming treatments, whatever name you give them. So these are the various types that are being practiced also, and they are literature is also cited on these. So this is a not a very new technique, but it does exist. Electron beam priming. Basically, it is being used as solution against the pathogens because the seeds are coated with electrons. So how is it done? There is a high electric voltage which is given between the cathode and the anode, and it results in the emission of electrons from the cathode. So these electrons which are emitted, they are accelerated. So they will come to an area which is called the process area, which will separate the place from where the electrons are being generated and the area where the seed is there and which is under ambient pressure. So the dose which is present on dose of the electrons which is present on the seed coat, it can be regulated by the changing by the strength of the current. So all these physical energies, one thing you should keep in mind, they are highly dose dependent processes. So a lot of standardization has to be done with the seed because the seeds will be at different moisture levels, the cultivar will be different. So all that kind of variation, some will be bold seeds, some will be small seed of different varieties. So all that standardization of the dose for how much time it should be given for like in magnetic field, we have to vary the field intensity also. So that has to be alternating current and direct current, and all those variables have to be taken care of before telling that or before coming to a conclusion that this dose is perfect for priming of this kind of a seed. Then there is the laser seed treatment. There are three kind of lasers, red, green, and blue, which are being used. This is an experimental setup from some lab. And here is a petri plate in which the seeds are being put and treated with the laser. And if you see the schematic diagram of it, this is the place where the seeds are kept and treated. Another technique is the cold plasma treatment. So in cold plasma, the water is activated, which is called plasma activated water, or directly the seed can be treated. So both the conditions have been shown to have very good results under experimental conditions. In fact, a few years, I think last year we had a seed company which was I mean, they wanted to have a collaboration with us for field trials because they are giving this technology to the farmers. It was somewhere, I think from a Gujarat, it was some company in Gujarat which wanted to have field trials with IRI. So because of COVID, such things are delayed. But maybe we'll look into the prospect of taking it again. So our group has been working for a number of years on magnetopriming, which is the treatment of seeds with static magnetic field. We all know that the earth, the place where we are sitting, has certain level of magnetic energy. So this energy throughout the earth's surface is very small, though you cannot ignore it. It is in the 25 to 65 microteslas. So at the equator, it won't be there, but as you move towards the poles, you will experience magnetic energy. And it was a simple observation which was seen in case of pine trees when they were grown at different latitudes. It was seen that the trees, the same species which was grown towards the poles, it had better growth and they were taller than the ones which were grown at the lower latitude. So it was not an experiment, it was simple observation which was correlated with that of the magnetic field. A very simple analogy which will be more comprehensible with everybody is the case of migratory birds. You must have heard about migratory birds coming to India from Siberia and returning back to Siberia in summer. So we change into products. If you see when the bird is facing towards this side, then the alignment of the reactant and the products is like this. So when it faces from east to west, or then the alignment is somewhat like this. So when these reactants and products are differentially aligned within the protein which is situated at the back of the eye of the bird, it can change its direction or it can move to the same direction depending on the reference where it has to be. So this is the unit which we have developed out of a BIRAC project, which is used for magnetopriming of seed. Maybe you can see it, it's just behind my table. So this is a big unit and each part is an independent unit. These are 3 parts. So if a farmer or a seed company requires to have more bulk treatment, they can add on to the parts. So this is a conveyor belt which is used. There are some non-magnetic boxes because this project was on vegetable production. So because vegetable seeds are low volume and high value, so and later on there was not much visible difference, and this led to a increase in the grain yield. So as I said, when I showed you a slide on the mechanisms, I told you that the ROS levels have to be maintained and also was signaling which was operative in the seeds which were magnetoprime. So with our collaborators at Devi Ahilya University, Dr. Guru Prasad's group, and Guru Prasad's group, they worked with different crops, and in this case it was soybean in which the embryo and the hypocotyl were removed after the seed was magnetoprimed, and they look for the ROS signatures. In this case they have seen the EPR spectra was seen, and it was seen if this is the ADAM simple phenyl N-T-butyl nitrone which is used. So the spectra of this chemical gave the signature like this. But if you see this was the control of the embryo, this was the control of the hypocotyl. And here if you see, the peaks became more prominent when the magnetic field was increased to 200 millitesla. This is for 150, this is for 200 millitesla, and this is the quantitative data of the same experiment. Similarly, that was for superoxide, this is for hydroxyl radical. Hydroxyl radical also you can see the signatures differing in respect to the magnetic field in case of the embryo and hypocotyl. So the plants which were raised from these treated seeds, it was seen that these reaction centers were found to be more in 200 millitesla treated seeds. This is a hypothetical model which has been generated from the data which has been taken on the crop. So this software generated model shows that there are more active reaction centers present in 200 millitesla. These dark dots, what you see, they symbolize that these are less, and in this control these are more closed. So more open ones are seen in case of 200 millitesla seeds, and the arrow also, if you see, this is thicker than that of this and the control one. This model was generated, software model was generated from all these parameters which were measured on the crop which was raised from 150 millitesla and 200 millitesla treated seeds. So it was seen that the photosynthetic performance of this kind of seeds, which is given by PS, is a performance index, it had improved for the magnetically treated seed compared to the untreated seeds. Next, in line with the physical energies is the microwave energy, where we have published 2-3 papers in which microwave energy was used to break the hard seededness of forage legumes. Forage legumes are known for their hard seededness, and that is why they have very poor germination. In case of this stylosynthesis, you see it is control has only 6.7% germination, and this seed lot had only 2.8% germination. So in developed countries, what is because they have huge pasture lands and grasslands. In certain areas, they used a big drum system to scarify to bring about the scarification of the seed. So how is it done? You must have seen a mortar at the construction site where there are huge drums in which the gravel and the cement etc. is mixed when the house or the building is being constructed. So it has an abrasive surface on the inside. So all the seeds are put in that, and due to the rotation of the drum, the seeds keep on in the drum, and finally the surface or the seed coat loses its integrity, and there is the germination process is enhanced. Germination of the seed is enhanced, means more will germinate. Sorry, it is not enhanced, more seeds will germinate. So we started in a very humble way. We had a microwave, domestic microwave, which we opened up, and we then an analog system fitted to it. We knew that we are giving this dose for this many minutes to this crop. And finally, out of the project, one was also generated for bulk treatment of seeds, in which this is a and here the seeds are put, and then there is a microwave generation in this area. The seeds will pass through it, and finally they'll be treated, and then they will be removed. So here you can see the percent germination from 6.7, it has gone up to 46% under this dose, and here also it has gone up to 70% under this dose. So we wanted to see what happens to the surface of the seed once these treatments are given. So we compared the untreated seeds with sulfuric acid treated, then hot water treated, then this was acid, and this was, I think, physically scarified with the sandpaper, and then microwave treated. So all these treatments, which was done under scanning electron microscope, the topography of the seed surface showed that in this case there were certain fissures or disintegration of the seed coat. It was due to the expansion and contraction of the seed coat under the microwave energy. Then very interestingly, there were these holes which were also created. These holes are present here also. These passages are present here also, but they do not open up. But in case of microwave treatment, like you pop your corns in the microwave. Similarly, these are certain polysaccharide accumulations which are present on the seed coat. So they pop up and they open up, and they allow the entry of water within the seed. Once imbibition is fast in these kind of seeds, then they will take in more water and germinate. So this was the process which was happening in the seed. Then we, through NMR, we characterized the extracellular water, cytoplasmic water, and then control we found. I would request you to notice this black bar. So in this you see it appearing at 64 hours, But in this case, the black bar appeared at 24 hours. It was a microwave treatment. So it's important because it will help in the hydration of all the molecules which are required for the germination process to occur. The microwave treatment, it helps in the weakening of the seed coat that you have seen through the SEM photographs, and it can also damage the heat sensitive inhibitors. Then it can cause the uh, cleaning of the capillary system which may be generated because of water molecule moving in and out, which can happen in case of seed coat. So once the seed coat loses its integrity, it becomes very natural for the seed if it is put under stress condition also. So whatever water is available under the, let us talk about drought stress, so that will become available faster to the treated microwave treated seeds than the ones which are not treated. So the not treated ones will require more water first for the seed coat to be softened and then disintegrated and then take up more water for further germination. But we have already created these channels or these fissures in the seed to more water to be taken out. There is something like stress memory which occurs in the prime seeds. So it is equivalent to upregulation for the gene which is involved and circulated. This resulted in protein accumulation and the germination was improved and so was the salinity tolerance. Now there is a stress memory. This was a stress memory which was created during the priming treatment and which was used later when the plant actually experienced stress or the seed actually experienced stress. So there are certain signaling pathways which are upregulated. Is the cascade which moves through the upregulation of genes or the downregulation of certain genes like ABA, GA, and ethylene, jasmonic acid. Then there are certain epigenetic changes which are occurring. So all these will be mediated through kinases. Then they can be expression of certain genes, downregulation or upregulation, to lead to the physiological response, which will be cell division, elongation, and then the establishment of the seedling. Now coming to the stress part of the physical energies, this is from our own experiments which we published in 2012. Here we created 0.2 megapascals of stress and 0.4 megapascal of stress. This was for 2 hours, this was for 1 hour. And this being a mild stress, if you see the turgor, this blue is 100 millitesla and this is the 200. In the mild stress, it was seen that 200 millitesla plants from the 200 millitesla treated seeds were showing higher turgor potential, higher relative water content, more leaf area, then shoot dry weight was more, the root dry weight was more, and of course the root to shoot ratio also was seen to be less in this case, but high in 100 millipascal stage, millitesla stage. This is from Sini's experiment, who is attending this meeting also. So in her work, she had used 80 millimolar sodium chloride, and the germination improvement what with increasing salinity, the 40 millimolars of sodium chloride salinity improved the vigor index to 21, and under non-saline condition, the magnetic priming resulted in 24%. Under saline condition, there was a higher increase, higher in the saline, though the values are less, but the increase was higher because priming, as you know, gives better results under stress conditions compared to the non-stressed conditions, be it any kind of priming treatments. So the root mass also, if you observe carefully, the root mass of the unprimed seed was less compared to the primed seed color compared to the green color. In the production of superoxide, which is an ROS, and nitrous oxide, which is an RNS, we compared these two. So these relative change, that is control minus stress, prime minus non-prime over the non-primed ones. These values are given here, and if you see, if the value is lower, that means the difference is less. If it is more, that means the difference is more. So here, if you see, these are the inhibitors which were used. This is for NR, and this is for NOS, and this is the nitrous oxide donor. So it was seen that under timing, under saline condition, though the change was less, but timing definitely led to more production of peroxide as well as nitrous oxide, and it also changed the level of ABA2 GA in case of prime seeds. And this we have done in collaboration with Dr. Sunita Kataria, who's also a dear friend from DAVV. And it was her experiment where we partly did some studies, and this we have communicated, and we are hoping. As I can't see anybody, I hope people are around. I was not talking to myself. Hello.
From the 40% increase in germination is also taking. I mean, treatments change that she has given a complete picture. And also, there is a stress study also in many of the experiments. And also, I have shown the profile of glycine, etc., which are really giving the stress tolerance to the plants. Then also I'm told about the stress basically being induced when there is a priming experiment, priming treatment. Also some evidence diseases, for example, notification probably the patient must also have it can be studied in the future experiments. And as people shown, for example, on the paper, she has done 10 plus then 0.2 megapascal express, 1.4 megapascal express with 100 and 200 milliliters. And being highlight that more sodium is better for our making the seed more minerals and the better eating in the field. Also, in the salinity studies where TT millimolar was used, here also the size percentage was there, and root mass percentage also even with the pictures of the root architecture and all and growth also included. The reason we're also to be explored and shown in a paper for the indole acetic acid, acid getting increased, abscisic acid getting picked down, and also the reactive oxidants species profile got changed, RNS conduct got changed, etc. And also in a collaborative paper, she has found about the arsenic stress related also the tolerance of data she has presented. One thing that personally to me is to do experiments uh, which was done by one of her
Yeah,
students.
that was not related to stress, so I did not present.
Okay, okay. So if you had that slicer there, it would have been more attractive. The tomato yield was increased. The tomato yield was increased. Growth and all. It was really. Also, ma'am, concluded the seminar with a few uh, back ahead. For instance, the research behind the market kind of prime should be really give understanding about the signaling pathways and probably an easier way to upgrade our seeds, upgrade our crops through magnetic priming, and also priming in general. And the interactome between the magnetic priming or any other priming experiment with the biomolecule profile, the biochemical profile, we have been studied on a real-time basis, which will give better understanding and opening up a very big arena for future experiments. And also, she mentioned about the cell membrane potential effects. Again, important because all the organs are there, then which in interacting with the magnetic effects, other priming effects, which in improving the and so with this, I would like to give the overall conclusion of what we learned today, and it was overall wonderful experience for us, and I hope we will be able to collaborate with you in the coming years, especially from the Seed Science and Technology Department from Kerala University. Thank you, ma'am. Thanks for your
Thank,
time.
thank you all. The honor was mine. Thank you.
Thank you, sir. Now, as we have come towards the end of the 10th lecture series or 10th lecture of the webinar series, I take this opportunity to thank one and all gathered here for the webinar. On behalf of the Department of Seed Science and Technology, I thank Dr. Anjali Anand ma'am for the detailed lecture on boosting stress tolerance of seed through physical seed treatments. It was a very informative session and we could learn so much about the different seed priming in detail. Thank you so much, ma'am. I also thank
Thank,
Dr. Krishnakumar sir for being a part of the lecture series and completing it with a concluding remark. I extend my gratitude to our teachers, Dr. Rosemary Francis, Dr. D.G. Bashing, and Srimati Fahida, ma'am, for bringing us this opportunity to attain knowledge from such experienced personalities. I also thank my colleagues and all the participants who has made this webinar a success. Thank you all.
thank you. Thank
Thank you.
you.